For those of you who don't know, Terry was my grad school and postdoc mentor. Terry got his bachelor's degree in biochemistry and microbiology from the University to get his PhD from Harvard with Rong Li, where he studied the mechanisms of actin assembly in budding yeast. And then he did his postdoc with Elaine Fuchs at Rockefeller, where he identified roles for desmosomes in recruiting centrosomal proteins and organizing microtubules and differentiated epidermal cells. And he also identified a developmental mode of regulated spindle orientation that the epidermis uses to stratify during embryogenesis. In his lab at Duke, Terry continues to build novel in vivo mouse tools to study epithelial morphogenesis, the relationship between stem cells and their differentiated daughters, and spindle orientation. While you can read all of these things on his CV, I wanted to personally comment on Terry's strong commitment to basic cell biology. He has instilled and continues to instill in his trainees a drive to follow the science while maintaining a solid understanding of the bigger picture. And he has taught me everything I know about science and American politics, even though he's Canadian. So thank you for coming, Terry. I'm proud to have you here. So, thank you.
Thank you very much. I'm very glad to be here. I think sometimes when we do introductions for the trainees, sometimes they hear all the good things and they never hear the bad things. And sometimes I think we should say how many grants we've had rejected or papers we've had rejected. But it only takes a few good choices in your career to make it through. And fortunately for me, Kaylin was one of those. So I'm very happy to be here. I had a great morning talking to a bunch of you already. I hope to see more this afternoon. And so I'm going to go ahead and share my screen. And today what I thought I would do is I would first introduce the lab really quick and tell you about the diversity of topics we work on, and then dive into one a little bit more deeply. So I'm really interested in sort of tissue architecture and how it's established, both at the cellular level and how the cells come together in three-dimensional patterns that mediate their function. And we study this really from some of the basic cell biological regulators, like what did the cytoskeleton do, what do cell adhesions do, what does spindle orientation do. But I was really trained as a basic cell biologist, and I love most the sort of nuts and bolts of the basic cell biological regulators and how these very conserved machines are used in different cell types to do very different biologies. And so today I thought I would tell you kind of a story that takes part of what I like the most, which is using tissue biology to understand basic cell biology, but then also to show you the opposite, how trying to study cell biology in a tissue context can teach us things about tissues that we didn't know before. And so I think the two of these stories go nicely together because they are kind of flip sides of that coin. All right, why is this not moving? Sorry. I don't know if my computer is frozen or what's going on. Okay, here we go. So I'm going to focus today on our work in the skin. And I just show this to highlight sort of the beauty and the complexity of the kinds of structures that the stem cells of the skin can make. And so we're all used to looking at skin and hair, but feathers, scales, horns, they're all derived from epidermal stem cells. And so trying to understand how these very functional and important structures are made is kind of an underlying goal. The skin itself is a little bit less pretty in this sort of model view, but I do want to highlight some of the important areas and the architecture just so we're all on the same page going forward. So our work is focused really on the epidermis, which is this thin layer of tissue right at the surface of the skin. It's separated from the underlying mesenchyme by a basement membrane. And you've got a single layer of progenitor cells that's on top of that basement membrane, and they're important for the renewal of the skin, the turnover, and any wound healing that has to happen. And then, because I've always had a great interest, not only in the stem cells, but cell biologically, I often think that the differentiated cells are much more interesting. And so these cells differentiate and go through a process of further differentiation, terminal differentiation, to form a barrier at the surface of the skin. Okay, and it's really important that this is a mechanically and chemically robust tissue. And so how you develop that architecture is really important. But beyond its sort, I think the skin is also a fantastic tissue to study from the perspective of cell and tissue biology. So obviously there's great genetic tools for gain and loss of function studies, as is true in many cases now, or many tissues. But as you can see in this embryo, the advantage of the skin is that it's right on the surface. And so we can image dynamics of the skin, and what we're really interested in is using it to study subcellular dynamics, right? And so over the past 10 to 15 years, we and others have been developing tools to ensure that we can visualize some of the major cell biological regulators in this tissue. And my lab has generated a bunch of these on top. This is by no means an exhaustive list. But I think one point that I do want to make is that, I've long been jealous of the toolkits that are available to C. elegans and Drosophila people. And I think we tend to be very behind in the mouse. And as the ability to image subcellular dynamics within living tissues has developed, I think it's important that we also develop the tools and the mouse strains that we can do this. And so it's kind of a goal of mine to continue to build on this toolkit in the future. And we can do things obviously like see structures. We can look at their dynamics, very similar to the ways that you might even image a cultured plate of cells. We can look at turnover, etc. And so in some ways, the mouse embryo is a really good petri dish for being able to do some of this cell biology in vivo. All right, so the projects I want to tell you about today are centered around the role of the microtubule cytoskeleton. And what I think, if you remember nothing else from this talk, I'd like you to remember the idea that in almost all differentiated cells in our body, microtubules reorganize into distinctive arrays. In many cell types, we don't really know what these look like. And in most cell types, we don't know what these do. But the textbook kind of diagram that everyone has for microtubules is this kind of diagram right here, where you have a strong microtubule organizing center or centrosome, and you get these radial arrays of microtubules that emanate from that point. But again, in differentiated epithelia and muscle and neurons, etc., these microtubules reorganize into cell type specific arrays, and they do specific jobs. And I will say that in very few cases, I think neurons being a great exception, do we have a really good sense of what the microtubules are doing in these differentiated arrays. Okay, and so we wanted to study this, and it turns out the epidermis is a great tissue to study this because it undergoes a very stereotypical reorganization of a microtubule cytoskeleton as cells differentiate. And so I just want to point out that when I show pictures of the skin going forward, they're always going to be oriented like this, right? So the epidermis will be up, dermis down, and then usually the basement membrane will be denoted with a dotted line or something. And so the basal cells are right on top and differentiated cells as you move up. And what we published a long time ago now is that in the progenitor cells, there is a centrosome. It acts as a microtubule organizing center, and you get these arrays of microtubules emanating out. But then as you differentiate, initially with initial differentiation, you get a very clear loss of centrosome MTOC activity. So you still have a centrosome, but it no longer acts to organize the microtubules. And then as differentiation proceeds, the microtubules become highly cortically enriched. And this is a very unusual organization for microtubules. It's actually a little bit reminiscent of what plants do, but both probably the mechanism and function are very distinct here. And so we've been using this system to answer a number of questions related to microtubule organization, dynamics, and function. And I'm going to talk about two of those today, which is how is the centrosome inactivated when cells differentiate? And this is a very common feature in neurons, in muscles, in epithelia, but in very few cases do we understand what the centrosome microtubule organizing center activity means. And I'm not going to tell you about our studies on how microtubules are reorganized or how their dynamics are controlled, but I'm happy to talk about those if people are interested. Excuse me. And then at the end of the talk, I want to talk about our attempts to understand really what the microtubular arrays do in these differentiated cells. And I'll tease you right now and say that I'm not going to answer this question. I'll tell you that we've been trying for a long time and it's led us down some really interesting paths, but I still think it's an interesting and open question. All right, so how is the centrosome inactivated? And all the work I'm going to tell you about today was done by two fantastic trainees. So Andrew Murayama was a graduate student in the lab who will be starting his own lab at UCSD this summer. And then Wenxu Ning is a postdoctoral fellow who really continued the work and took it in directions that I never expected to go. All right. And so the first question that actually Andrew had to answer in trying to address this question is, are there really changes in the centrosome itself, right? Because you can imagine that what's happening here is that other cellular sites just become very good at organizing microtubules and they outcompete the centrosome. And so the question was, is this a centrosome intrinsic change or change elsewhere? And to address that, Andrew developed methods to isolate centrosomes from both proliferative and differentiated keratinocytes in culture. And this was really a modification of protocols initially developed by Tim Mitchison's lab. And so then once you isolate those centrosomes, you can mix them with purified tubulin in an assembly buffer, let them assemble, fix them and pellet them onto cover slips to ask, do they form asters? And what you can see here is that the centrosomes from proliferative cells form really robust microtubule asters, and those from differentiated cells do not, okay? And so it really is a change occurring at the centrosome as the cells differentiate, okay? And so we want to molecularly understand what the changes were in the centrosome that caused this change in its activity. And I'm not going to go into a lot of depth, but the centrosome is a complex organelle-like structure, non-membrane bound. It's built around a core of two centrioles. And then for our purposes, what's important is that there's this mass of pericentriolar material, which contains hundreds of proteins, which contribute to its architecture and its function. Okay, but most important, or one of the most important of these, is a protein called gamma-tubulin, which is a component of the gamma-tubulin ring complex, which is both highly enriched on centrosomes and the best characterized microtubule nucleator in cells. And the other thing that you're going to need to know for this talk is that there are a couple of proteins that are thought to directly bind to gamma-tubulin and to tether it to the centrosome. And these are proteins called NED1 and CDK5-RAP2. Okay, so Andrew noticed when he just started looking at gamma-tubulin in the skin is that in the progenitor cells, in these basal cells, it had very strong enrichment at the centrosome. But even in the cells sort of one layer above that, who have begun to differentiate, you see that the levels at the centrosome are way down. I'm actually going to show you that in cultured cells because I think it's a little bit easier to see if you're not used to looking at the tissue. So we looked at a series of centrosomal proteins in both proliferative and differentiated cells, and you can see that gamma-tubulin is quickly lost when cells differentiate, as is this interacting protein NED1. But most of the other proteins that we looked at that are part of the pericentriolar material remain and don't change upon initial differentiation. All right. The way that these proteins were lost, though, is actually pretty distinct from each other. So when we looked at whole cell lysates under these conditions, what we found is that the levels of gamma-tubulin remain constant. So the protein is losing its centrosomal localization, but it's still present in the cell. NED1, in contrast, is pretty rapidly degraded, and so it's gone from the cell. And that offered a pretty clear hypothesis, right? That NED1 is recruiting gamma-tubulin to the centrosome, and that in the absence of NED1. And so we could test that hypothesis pretty easily because we can knock down NED1 and or CDK5-RAP2 in these cells and ask what happens to the centrosomal gamma-tubulin levels. And consistently, I'm not going to show you the data, but for both of these proteins, we get over 90% knockdown. And we have to do this transiently because NED1 is important for mitosis as well. And so you can see that NED1 is essential for recruiting the majority of gamma-tubulin to the centrosome, okay? And what was funny or noteworthy is that CDK5-RAP2, which is this other gamma-TURC binding protein, was responsible for recruiting just a small fraction of the centrosomal gamma-tubulin, okay? So the really surprising finding for me was when we looked at the nucleation rate of centrosomes in living cells. And what you can see here is that even though we've gotten rid of 80% of the centrosomal gamma-tubulin, we have almost no change in the nucleation rate at the centrosome. And we measure this by looking at EB1 puncta that emerge at the centrosome and grow out from that site, okay? And that suggests that the NED1, the gamma-TURCs recruited by NED1 are not significantly contributing to the nucleation of new microtubules at the centrosome. In contrast, in the CDK5-RAP2 cells, even though we've only lost a small fraction of the gamma-TURC, we see a pretty substantial decrease in centrosomal nucleation levels, okay? And I think the important result from this is one, that we can't use gamma-TURC or gamma-tubulin as kind of a surrogate marker for nucleation in the cell, okay? It's a little bit more complex than that. And this, to us, also suggested that there are two different gamma-TURC complexes at the centrosome, one that's bound to NED1 and one that's bound to CDK5-RAP2, and that is the one that's really important for nucleating new microtubules. Okay. So in support of this, we wanted to sort of be able to reconstitute these complexes and understand whether their in vitro activities really matched up with what we're seeing in vivo. And so Andrew isolated gamma-TURC complexes and then mixed them either with fragments of NED1 or CDK5-RAP2 that are important for binding and interaction. And in work that was in agreement with previously published work that CDK5-RAP2 can activate the nucleation activity, we saw a substantial increase in microtubules on the cover slips when CDK5-RAP2 was present, but the NED1 interaction domain didn't affect the activity at all, okay? All right, and also we found that if we isolate the centrosomes from NED1 knockdown cells, they completely recapitulate the phenotype that we'd seen before in differentiated cells, okay? So just getting rid of NED1 makes these cells really poor microtubule organizing centers, okay? And that's quantitated here. So but these assays can actually tell us a little bit more, because the way that we do them is we do the assembly in solution and then pellet them onto cover slips. And so we can look at the centrosomes and the asters surrounding them, but we can also look at the free microtubules that have been nucleated in solution. And our surprise there is that these guys are really poor MTOCs, but they're almost as, or just as good as the proliferative centrosomes in nucleating the formation of new microtubules, okay? And so again, this suggested that there's not a defect in the ability of these cells to make microtubules, but there is a defect in the ability to stably tether or anchor the microtubules to the centrosome, okay? And so together, this work suggested that there's really two distinct types of y-TURCs, both localized at the centrosome. One that's bound to CDK5-RAP2, and it's really important for microtubule nucleation. And then another one that's bound to NED1, and we're suggesting that this is a complex that has a distinct function in anchoring the microtubules to the centrosome. And that is based on the following kinds of experiments. So I'm just going to show you one example. So what we decided to do was to move this machinery to a different location in the cell that's normally not competent to either nucleate or to anchor microtubules, okay? And we did this by targeting these and to desmosomes specifically. And what you can see is that putting either of these proteins alone at the cortex does not cause a really significant reorganization of the microtubule cytoskeleton, okay? What I'm not going to show you here is that in the case of CDK5-RAP2, when you have this at the cell cortex, it's actually enough to stimulate nucleation from the cortex. And so you get a really non-physiological situation. We're getting microtubules anchored at the cortex and moving into the cell, which is the opposite of what normally happens. But when we have both of these proteins together at the cortex, we see that we get stable anchoring of microtubules at those sites, okay? So we can not only nucleate the formation of new microtubules, but they're stably anchored here, okay? And this was important because when we think about cytoskeletal organizing structures, whether they be for the actin cytoskeleton or the microtubule cytoskeleton, those structures have to, or in general, promote not only the assembly of new polymer, but they stably associate with that polymer, okay? And this is an activity that has remained largely unexplored for the centrosome. And so this suggests that one of the ways that centrosomes are able to stably remain associated with the microtubules that they nucleate is through these NED1 y-TURC complexes. So, and then really cool, just because it's cool phenomenology, Andrew found that when these cells went into mitosis, that the cortex actually began to act like a spindle pole, and you would get these really messed up mitoses where random parts of the cortex would be pulling on the chromosomes and trying to segregate them away, okay? So I think this was a really great visual example of the ability of these domains to act together to really organize the microtubule cytoskeleton. All right. So in work I'm not going to tell you about, we were also interested in the regulation of this activity. So when the cells undergo differentiation, they lose MTOC activity, they lose centrosomal NED1 and gamma-tubulin. But what drives that process? And we spend a lot of time looking at differentiation factors that regulate this pathway in the skin, especially Notch, because it's kind of thought to be, at least in the embryo, a major determinant of differentiation. But it turns out to be a lot more simple than that. And it's simply the entry into quiescence that drives this process. And you can mimic it simply by inhibiting cell cycle CDK kinases. And I think it's kind of an elegant way for the cell to couple loss of centrosome activity to full differentiation, right? Because they're not going to have to recommit to another mitotic cycle, so they can lose MTOC activity. And I should say that we kind of almost think of this as a rheostat or a dimmer switch, right? So when you go into mitosis, CDK levels go up, right? And this activates the centrosome to become a spindle pole. But similarly, when you enter quiescence, you also can dial that low level of CDK1 activity down, and that shuts off the centrosome. All right. All right, so that's an example, I think, and one that I'm really proud of, where trying to understand what's happening in the tissue taught us lessons about some basic cell biological regulators. And so for the rest of the talk, I want to talk about this question number 4, which is, what are the physiological functions of these cortical microtubule arrays, and how do we get at them? And I should say that we tried this from a few different perspectives. And we tried to start out by doing it rationally, understanding the proteins that reorganize the microtubules, knocking them out, trying to understand the phenotypes. But we kept running into this problem in that these proteins that we identified really had pleiotropic functions and things like cell adhesion that made it really difficult for us to interpret the results. And so we decided to start with a really blunt and sledgehammer approach, and that is to ask what happens if we just get rid of all the microtubules in these differentiated cells. Okay, and for this experiment, it's really important that we do this specifically in the differentiated cells, because obviously getting rid of microtubules in a mitotic cell is going to lead to mitotic catastrophes and will lead to uninterpretable results. So Andrew had to design a couple of different mouse strains to be able to do this. System to allow regulated expression specifically in differentiated cells. And he did this using a K10 promoter driving RTTA for doxycycline inducible expression. And then the second component, and this is just showing that you can get robust expression specifically in the differentiated cells and not in these progenitor cells here in the embryo. And then Andrew paired this with a transgene that expresses an active form of spastin. So spastin is a microtubule severing protein that can really have very different effects on microtubules depending on the conditions. In our case, we're using an activated form of the spastin gene, which I'm not going to go into detail here, but is really good at clearing out the microtubules from the cell. And when Andrew put these two together in the mouse, he found a result that was incredibly surprising for us, in that this is what the skin looks like in a control mouse. And after 2 to 3 days, where we're getting rid of the microtubules in the differentiated cells, he sees this really dramatic change in the tissue architecture. Okay, and so I will tell you that this effect is actually very cell type dependent. We've now done this in a few different cell types. And in cell types where I thought microtubules were going to be really important for cell shape and physiology, like the very tall columnar cells of the intestine, they actually have very subtle roles. And in the skin, where going in, I wasn't really sure whether they'd have much of a role at all or not, they really had a profound effect. And so we're really interested in what's driving this really dramatic change in the architecture of the epithelium. And there's 2 big answers to the question. So first of all, the epidermis is a stratified squamous epithelium. And squamous just means that as these cells differentiate, they get really flat, okay? So you can see an example of what a cell looks like here. In these differentiated granular cells in the mutant, we see that this squamous shape is lost and the cells remain more cuboidal or circular, spherical, okay? And so obviously this is contributing a large amount to this, because if the cells can't flatten, the tissue is going to be much thicker. But by itself, it can't really explain that, because there's also more cells present. And the other part of this equation is that when we get rid of the microtubules in the differentiated cells, we see an unexpected non-autonomous response in the progenitor cells that underlie them. And we see a really strong increase in proliferation of these cells. So this result, I still think, was the biggest surprise to us, because we did not really think of the differentiated cells as being part of the niche that controls progenitor cell behavior. We kind of view it as a one-way track. You go from progenitor cell to differentiated cell, and that's the flow of information. There's been previous data that the progenitor cells can respond to the physiology of the tissue. So if you disrupt barrier function of adult skin, you'll often get responses including proliferation of progenitors. But what we're seeing here is not related to the physiology, and it actually occurs even before a barrier forms in this tissue. And so we're really interested in this idea that the differentiated daughter cells can regulate the behavior of the underlying progenitor cells, and wanted to understand the mechanism underlying that. And so this is work by Wen-Chu Ning. And what she did is she isolated the differentiated cells from the control in mice, and she profiled them by RNA-seq. And among the differences that we noticed, we saw that there were strong changes in the actin cytoskeleton and in Rho-GTPase signaling, which many of you will know are major regulators of the cytoskeleton and contractility. And more specifically, there's a group of genes called the contractome, which were just identified or classified as genes that have been implicated in regulating actomyosin contractility. And we saw that almost all of these were differentially regulated in this tissue, including upstream Rho-GTPases, intermediate kinases, and even myosins themselves. Okay. So to validate these results, we went back to the tissue and asked, what's happening to. And you can see here, both in the images and the quantitation, that there's a clear enrichment of cortical actin and also cortical myosin 2C in these mice. And not only that, but if we actually look at the mechanics of the tissue, we can take pieces of the tissue and culture them in vitro for a day or two. We see that when we get rid of the microtubules, these guys are much more contractile and shrink. And when we see cortical actomyosin, our first thought is always the adherence junctions, okay? Because the adherence junctions are the cell-cell contacts that bind to the underlying cortical actin. And we know that that tension on that actin can induce conformational changes in components of the adherence junction. So one of these is a protein called alpha-catenin, which exists in different conformations under low and high tension. And under high tension, we have a nice tool because there's a conformation-specific antibody called alpha-18 that will preferentially bind to the form of alpha-catenin under high tension. And what we see in these mice are that while the levels of alpha-catenin at the cortex don't change, which suggests that they don't have more adherence junctions, the level of alpha-catenin within those junctions is increased, consistent with them being under more tension. So junctional tension can actually have very diverse effects on junction dynamics. There have been cases where increased tension has been reported to stabilize junctions and to destabilize junctions. And to understand what the effect was in this tissue, we turned to fluorescence recovery after photobleaching assay. And for this, we used an E-coherent CFP knock-in mouse that was generated by the Cleavers lab. And what I hope you can see here is that there is a really substantial stabilization of the adherence junctions when we get rid of the microtubules, okay? Consistent with contractility stabilizing these junctions. And we'll get back a little bit later to why we think that might be important for some of the phenotypes that we're seeing. All right. So this suggested that one of the effects of getting rid of microtubules is increased actomyosin contractility. And we want to know, is this increased actomyosin contractility responsible for any of the phenotypes that we observe? And so we can test this pharmacologically by hitting the pathway at different points with inhibitors of either the ROCK kinase or of myosin directly. And we think that these inhibitors are working as we expect in the tissue because we can see loss of this alpha 18 signal. And so we can rescue back to sort of wild type levels of contractility. And what we find is really a dramatic rescue. If you just want to look here on the bottom. So here is the mutant, and then you can see the thickness here. And then treatment with either Y-27 or blebbistatin causes rescue to essentially normal levels of tissue thickness. Okay, but importantly, it also rescued a lot of the cell shape and also the proliferation rate of the progenitor cells. Okay, so not only are the cell autonomous effects of microtubule disruption rescued by actomyosin inhibitors, but so are the non-autonomous effects. Okay. Because these mice now have this combination of things going on, right? They've got loss of microtubules, of increased actomyosin. We wanted to know whether the actomyosin contractility was really driving these phenotypes on its own, or whether they were working together with the microtubule phenotypes. So to ask that, once you developed another mouse strain in which she can conditionally express an active form of a ROGEF to essentially just drive this pathway under our control. Okay, and we're going to really quickly go through the results because they essentially mimic in every way the effects of getting rid of microtubules. So we see the same expansion of the tissue. We see the same contractility. We see the same cell autonomous changes in cell shape. And we see something I didn't discuss before, but in these animals. And we also see the effects on junctions, increased tension, and decreased turnover. Okay, so that was all good. But what was also great to see is that all the non-autonomous effects were also conserved in this mutant. Okay, and so we're again seeing an increase in proliferation in response to this, and we're seeing, for example, nuclear localization of the mechano-responsive transcription coactivator, or YAP. One thing I'll point out that I think is really interesting is that we normally think of YAP as being expressed in progenitor cells and driving their proliferation, but we actually noticed that differentiated cells as well have nuclear YAP. We're really interested in whether there are distinct roles for YAP in differentiated cells in mediating this response as well. All right. So similar to the case where we were getting rid of the microtubules, we found that we can rescue also the proliferation phenotypes in this background by various inhibitors of contractility or of YAP itself. Okay. And so what I'm very excited by sort of going forward is trying to tease out the roles of mechanical and chemical cues coming from these differentiated cells and how they impinge on the progenitor cells. And I'll discuss that a little bit more at the end of the talk. But I want to switch gears for a moment because we also observed a really another unexpected phenotype for me in these mice, and that they had a lot less hair than their litter mates. And one thing I want to stress going forward is that the cells that make the hair follicle are not being directly affected by our transgene. Okay, so again, we're seeing a non-autonomous effect here that's causing loss of hair. Okay, and this is just shown in section. All of these green spots here are hair follicles. You can see that there's a lot fewer. Okay, so we want to understand why there's a defect in the ability of the progenitor cells to form these hair follicles. Oh, and before I do that, I should tell you also that similar to the proliferation, we can rescue to a large extent this phenotype by decreasing contractility. So the increased contractility of these differentiated cells is somehow affecting the ability of the progenitors to grow into hair. All right. The formation of a hair follicle has been really well studied, and we know a lot of the intermediary steps in the initial formation of a placode, which then goes on to form a hair. And for your purposes, all I want you to know is that within this layer of progenitor cells, they're mostly going to contribute to making the interfollicular epidermis. Some of them are fated towards hair follicle cells, and the earliest way that we can mark these is LEF1 activity and Wnt signaling, okay? Subsequent to this, these cells will coalesce and begin to turn on subsequent transcriptional regulators and other factors that drive their further morphogenesis, okay? And so we label these as kind of early, mid, and late stage. And what we found early on is that when we look at any of these mid or late stage markers, they were always consistently decreased in either the K10-spastin or the ARH-GAF mutant, where we increase contractility. But if we look earlier at the initial specification, again using Wnt1, and now we're looking, instead of in cross-section, we're looking at whole mouse looking straight down on the skin. So each one of these little circles is a placode that will go on to form a hair follicle. We can see that while the late markers like LHX2 are missing, the early markers are there, okay? Oops. And so what we're seeing is not a problem with specification, but an inability of these early placodes to go on and form the late placodes and form hairs. And to understand why that was happening, we turned to live cell imaging. We start dye or floor. And what you can see here, I hope, is that over time, these loose placodes begin to tighten up. And you can quantify that by just looking at the diameter of their structures over time. You can see that in the mutant, these changes don't occur. So they stay really at the same size as when they were initially formed. So using a nuclear floor, we could follow the dynamics of these cells. And what we noticed is that, again, they didn't progress, and that the movements of the cells was decreased in all these cases, okay? And so our model is really that these cells can't move to form the tight placode that's needed to form to really amplify the signals and set off the downstream signaling required for the morphogenesis, and then they disappear. We're really interested in the idea that this is maybe not really due to differences in contractility or pulling forces of the cells above them, but it may be actually directly related to the stabilization of the adherence junctions between these cells, right? So these cells need to be exchanging neighbors with each other and with the cells on top of them as they migrate in. And we think, or one of the models is that the inability to remodel junctions is preventing this migration. And so one way to sort of begin to think about that is to look at other cells that migrate through the epidermis. And at this time during development, there are neural crest rod cells that become melanoblasts, and they invade up into the epidermis, and they're really rapidly migrating through. And very different from the migration of keratinocytes, these guys put out long projections and then move their nuclei through them. And what we found is that these guys have no problem putting out these projections that can weave around the keratinocytes, but they can never really move their nuclei through those paths, right? And so we think that that requires a lot more remodeling of the cell-cell contacts, and therefore they're kind of stuck and they can't push through the other keratinocytes. All right. So to sum up, what I've told you is that our work trying to understand the roles of microtubules in differentiated cells has not really answered that question, but it has told us about really unexpected roles for the differentiated cells in controlling many aspects of the behavior of the tissue, including proliferation, commitment to hair follicle fates, and migration. We think that this has really important implications for how we think about tissue biology and how we study it, right? And so for most of us who work on stem cells, the way we do this is we recombine genes within the basal cells or the stem cells, and we ask, well, what do the stem cells do now? Okay, but if you have a quickly turning over tissue, like the ones that my lab works on, the skin and the gut, it's really hard to find a time point when you've gotten rid of these proteins in the stem cells, but not in their differentiated daughters, right? And so I think we have to be very careful as we analyze those data to really understand, are we looking at cell autonomous effects due to loss of proteins in those cells, or are we actually looking at non-autonomous effects due to changing the niche of these cells? And as daughter cells are part of the niche, I think that becomes a real possibility. Okay, so my lab is very interested in sort of developing tools to begin to work through this problem, right? And I think that the real problem in this field for us is that the perdurance of mRNAs and proteins in these rapidly turning over tissues is such that we can't use traditional Cre-recombinase type ablation studies to distinguish roles in one type or the other. And so, inspired by some of the work in flies and worms that has targeted proteins instead of DNA, we've turned to these Degron systems, which offer the advantage of being faster and allowing loss of the protein, even if the mRNA pre-exists. And so the way these work is essentially, either through an antibody type interaction with a tag, and then that's linked to ligase machinery for ubiquitination and degradation. And so we screen through a bunch of these types of nanobodies to look for ones that would work well, found ones that worked well in culture, and we've now gone into the mouse, and we can see actually really robust loss of protein. So what I'll tell you here is what you're seeing on the outer edge here is autofluorescence, but the GFP signal within these cells is essentially completely gone. And so we're really excited by the approach in pushing this machinery to a level where we can do loss of function with it. And we're especially excited because if I go back to sort of the start and I talked about the toolbox that we try and make, right? The big roadblock in this is it's really expensive to make mice. And especially if you want to do, if you're interested in a gene, you're going to want to do loss of function in addition to being able to visualize the protein. And so our hope is that this kind of technology will allow us to build a single mouse that will allow us to both visualize the protein and its localization and dynamics, but then also to target it for degradation and do loss of function studies. And so far we've been really impressed by the kinetics of this and its ability to target sort of diverse proteins. And so fingers crossed that will lead us in new directions. So I'm going to end there. I really want to thank the entire lab, especially through this last year of, not being able to have the kind of environment that we want, but to keep working and doing the great stuff that they do. So all of the work I talked about here, I think, was done by both Andrew and Wenshu. But I've been incredibly lucky throughout the course of my lab to have really fantastic students. And so I will end there. I will thank funding sources and collaborators and all of you for listening. And I'm happy to take your questions.
Thanks, Terry. So I guess I should encourage trainees to ask questions first. You can raise your hand or put it in the chat. I don't see any hands raised, but there is a question in the chat. I can read. It's from Junlu. He said, why decreasing microtubules leads to increased actomyosin activity and contractility? And is the reverse also true, that decreasing actomyosin contractility augments microtubules?
Right. So those are both great questions. So there is precedent for getting rid of microtubules, increasing actomyosin contractility in a couple of different cases. And in those cases where the molecular mechanism has been somewhat addressed, what's best studied, I think, is this GFP-H1, which is a microtubule binding GFP that tends to be inactive when bound to microtubules, but when it comes off of them, it becomes more active and so sets off actomyosin contractility. We are testing whether that is relevant for this system. I think it may be a little bit more complex, right? Because in those systems, it's largely thought to be a non-transcriptional mechanism. And what we don't yet know is in the skin, how much of this is transcriptionally driven and how much is not. Because obviously we initially discovered it because all this contractility machinery is upregulated. And so that's one thing that we're trying to understand really is, is that a self-perpetuating pathway, or is it just the contractility pathway, or just the transcriptional pathway or non-transcriptional pathway that drives it? And I should also point that this is a very cell type specific effect. So if we get rid of microtubules in the differentiated cells of the intestine, they do not become hypercontractile. So different cell types will respond differently to this type of perturbation. And then the second question was, is the reverse true? If we get rid of contractility, do we increase cortical microtubules or something like that? And I don't have an answer for that. We've not looked under cases where we've decreased contractility to see that affects microtubules. But I think, it's an interesting question whether there's crosstalk both ways.
And then Valerie Horsley has a question. Do you think the differential desmosomal coherent expression in the differentiated cells impacts their ability to
do I think, so I can't totally answer that. I'm going to say my guess is no, for this reason. So one phenotype that I did not tell you about in these mice is that increased, and this is really surprising to us, and we're very excited to understand why this is, that increasing contractility actually causes desmosome loss. And it does not cause loss that's sufficient enough to cause tissue disruption, but it actually will if you perturb the adherence junctions. Then the whole tissue rips itself apart because it has nothing holding itself together. So I would say that I don't think we can rule out a role for desmosomes, but because of the connection between adherence junctions in the actomyosin cytoskeleton, we've been much more interested in the roles of both the stability of the junctions and mechanical signaling through the adherence junctions in regulating that. But I honestly, I can say that we have no good way to rule out a desmosome effect as well.
Okay, I'll ask a couple of questions. So my first question is a technical one. Is your Degron system, how do you induce it?
It's doxycycline, it's a tree.
That's tree.
Yeah.
So one thing I've struggled with, since I've known about this work is with this increased contractility, but a premature barrier formation, is how much do you think is actually a contractility problem and how much is a differentiation problem? And can you actually tease those two things apart?
Well, so I can't say that like in the adult, you're not going to get some of it like over extended times. you might have some contribution from a barrier. What I can say is in the embryo, we see the effect before there's a barrier issue or even a barrier that exists. And I can say that even in the adult, so we start to see proliferation 12 to 24 hours out kind of thing, right? And at those points, we're not seeing any of the changes in tissue, right? I mean, I guess you can argue, is it possible that there are already barrier defects, you know, in transepithelial water loss or things like that? I think that's possible, but honestly, you don't get the kind, we've never seen this kind of really robust proliferation in response to a barrier defect, right? I mean, you'll get increased proliferation, but not to this sort of level.
And then, sorry, one more question. Have you been able to look at turnover rates? Like, do the cells turn over less? Like, is the sloughing rate, I guess, decreased? Like, are the cells just longer lived too?
I don't know the answer to that. So I think that's a really interesting question. And there's actually someone who's coming, postdoc, who's really interested in understanding corneodesmosomes and sort of the mechanism by which cells slough and which you get rid of these corneodesmosomes that hold the squames sort of on the skin. I think it's a really interesting and really or pathologically relevant question, right? Because in human disease, you know, in these hyperkeratotic conditions, you see like often this really dramatic expansion of the cornified envelope and defects in its structure. So we haven't looked at that, but I think it's sort of a really interesting thing, like how the skin regulates the thickness of this dead tissue and how it controls its disassembly and stuff like that.
Thank you. Does anyone else have any questions? All right, well, thank you again, Terry, for coming and sharing.
Thank you, everybody.